The neural basis for pain during labor. This is a work done in Chang'an University and Chang'an Memorial Hospital, Taiwan. My name is J.J. Wang. This commentary was made on the article published in Brain Imaging and Behavior 2020 by Yang et al. Entitled Functional Human Brain Connectivity During Labor and its Alteration Under Epidural Analgesia. They recruited 15 parturients in the active stage of laboring and scanned them by using fMRI. They were divided into two groups, with and without pain relief by epidural analgesia. The study began after IRB was approved. Enrollment of the study subjects were at the outpatient clinic. They were prepared in the pre-delivery room following the standard procedure, and were sent to MRI room for following fMRI examination. During the MR scanning, they were carefully monitored using the squeeze ball. We monitored their oxygenation and respiration. And they were given a response pad, which they indicate the beginning and end of their uterine contraction. A study nurse followed the parturients into the scanner room and monitored the life signs and everything carefully. The parturient was scanned for approximately the next 30 minutes. The images were processed in the software statistical parametric mapping. A brain activation map was created. The experience of pain varies with individuals according to their physiological and psychological states. As a result, it is a multi-dimensional experience that involves several neural systems. For example, sensational and perceptional, or affective and modulatory. The sensory network consists of the primary and the secondary somatosensory cortexes. The affective system might involve anterior cingulate and the prefrontal cortices. The modulatory network might involve the medial and the frontal cortices, while the insula serves as a convergent zone for components in the pain network. The preliminary result shows fMRI of a woman in labor without an epidural analgesia. The experience can induce significant widespread changes in the central nervous system that is consistent with those reported from either gynecological or visceral pain. For example, lentiform nucleus and insula. The experience of pain during labor is unique because it is spontaneous and is exempt during the active state. This study showed extended involvement of the cingulo-frontal regions, which might exert a top-down control over the sensation and affection of pain. This network can be altered after the application of epidural analgesia. The involvement of neural networks and the cortical-to-cortical connections may be modulated. The involvement of anterior cingulate and the prefrontal cortex might suggest the achievement of pain relief by efficient control over the pain network. Because anticipation from the parturients might contribute to the effect of pain relief, the effect of epidural analgesia on the component in this multidimensional neural network will require further investigation. The figure shows the connectogram from two groups of parturients. In the left-hand side are those with epidural analgesia. In the right-hand side are those without any pain relief. The connectogram shows the significance of the correlation between cortical regions which were color encoded. We can notice that significant alterations of the connections throughout the brain. This preliminary study affirmed the existence of a brain signature for pain experienced during labor and show that this signature can be altered by the administration of epidural analgesia and can be accompanied by alterations in functional connectivity, most prominently observed in the cingulo-frontal system. It would contribute to the important advances in maternity care by conducting a more refined model of effective connectivity analysis, which reflect how brain areas crosstalk in obstetric pain. Inhibition of anterior cingulate cortex neurons might represent a potential new therapeutic target in alleviating labor-associated pain. For any further inquiry, please refer to the following email. And thank you for listening.